Today we're going to talk about biosimilars. So we'll be talking about generic drugs, we'll be talking about biologics, and we'll be talking about biosimilars. So that's our goal for today. Let us imagine a factory, let's say. Okay, so let's call this factory A. Okay, this factory is producing a well-known drug, say drug A. Okay. So this company is the first, let's call it a pharmaceutical industry. Company. It produces a new drug. A new drug, maybe it's targeted at a novel pathway. Okay, maybe the scientists here, they looked into the literature, research from academic institutions. Let's say published research, okay, which talked about a novel pathway for a disease. And they set out to produce a drug A targeted at this novel pathway that gives a therapeutic effect. Okay, it's beneficial. When we say therapeutic effect, we mean it is beneficial. All right, so this company, because it produced the drug first, this is called an innovator company. Okay, so they produce a drug A. And what is the input to them? They spend about $3.6 billion US dollars, and they take about 10 to 15 years to get this drug approved. Okay. So, a pharmaceutical company has got investors. Who invest money in the company. And why do they invest money? To get a return. Meaning they expect a profit. Right? So this company is supposed to market this drug and generate a return of investment to the investors. Okay. So they have put in $3.6 billion and they've put in this much of time, and then they start marketing it. Okay, so to market it, they have to price it. So they price it. So how do they price it? They look at whether it has to be affordable. At the same time, it should generate a profit. Okay, so that is how the market works. Now, in this process, they also patent drug A. They patent drug A. And the life of the patent is 20 years. Okay. So they hold the right to exclusively produce this drug and sell for 20 years. So the assumption is, in this 20 years, they have generated enough return of investment to let go of the patent. So when the patent expires at the end of 20 years, so the patent expires, okay, after 20 years. Okay. Which means the field becomes open to other companies. Companies to manufacture this drug. Okay, so after the expiry of the patent, if other companies manufacture this drug, it is no more the trade name drug, it is called generic. Okay. So now we have told that there is this company A which has spent 3.6 billion USD, and how many years did it take? About 10 to 15 years. Okay. And maybe there is a company B that wants to make a generic version of this drug. Okay, but the problem is, this company does not have 3.6 billion US dollars. Okay, and they also cannot afford to work for 10 to 15 years. Okay. So the idea is, this company, even with limited resources, should be able to produce this drug without spending 10 to 15 years. So the regulatory pathway is called is an abbreviated pathway. Okay, so what does abbreviated mean? Abbreviated means all the tests needn't be done. Okay, so the idea is, when a company makes a generic drug, the API, or the active pharmaceutical ingredient, the active pharmaceutical ingredient is same. Okay. So the regulatory authority says you can get an approval very fast because it is a generic drug. You need not prove safety and efficacy. Okay. You can use the data for safety and efficacy from the innovator company. Okay, so the data of the innovator company can be used to file a, a generic for producing a generic drug. So as a result, this second company B needn't do all these studies. So then it is called abbreviated. It is not the full pathway, but it is an abbreviated pathway. And what is the advantage to the consumer? For the consumer, the benefit is that company B does not spend 3.6 billion US dollars, and they don't take, how many years did we say? 10 to 15 years. They don't take 10 to 15 years to produce a drug. Okay. So as a result, because the investment by the company is less, they can afford to price the drug lower. Okay. So the lowering of the price does not imply reduction in quality. Okay, because the quality is assessed by the FDA. They cannot compromise on the API. The API should be absolutely the same. What will be different is maybe the excipients. So what are excipients? Excipients are things which are not API but are required for formulating. Okay. So the generic drug will be different from the brand name drug, maybe only in the excipients. So from the regulatory standpoint, company B should prove bioequivalence of their drug with that of the innovator. Okay, they have to prove bioequivalence. So if bioequivalence is proved, if bioequivalence is proved, then the generic drug becomes therapeutic, becomes therapeutically equivalent, drug. Okay, meaning these drugs can be substituted at the level of the pharmacy without the consent of the prescriber. Okay. Can be substituted without the consent of the prescriber. Okay. So generic drug should be bioequivalent. So generic drugs should be bioequivalent. They should contain the same API, the same active pharmaceutical ingredient. So that becomes a generic drug. So here when we say a generic drug, we mean a small molecule drug. Okay, so we mean a small molecule drug. So what is a small molecule drug? A small molecule drug has got a molecular weight less than 1000 Dalton. Okay. So now the question is, what about large molecules? Okay, large molecule drugs. What happens when the patent for these large molecules expire? Is there an abbreviated pathway similar to small molecules? Is the question that we are going to consider. So what is a large molecule? What is a large molecule drug? A large molecule drug has got a molecular weight greater than 1000 Dalton. Okay, they are also known as biologics. Okay, they are also known as biopharmaceuticals. Okay, they are also known as biologics and or biopharmaceuticals. Okay, they are also called biotechnology derived drugs. Okay. Now, they are produced by bioprocess. Okay. So they are biopharmaceuticals which are produced by a bioprocess. So what is bioprocess? So bioprocess is utilizing a living system to produce a biopharmaceutical. So now the question is, what is a living system? A living system can be cells, they can be tissues, they can be organs, or an organism itself. Okay. So what type of cells are we talking of? These cells can be, say, bacterial cells. Okay. So there are certain strains of bacteria. There are certain bacterial strains, commercial strains like called origami. These are all trade name. Okay. There is another called E. coli, shuffle. Again, that's commercially available strain. It can be, let's okay, it can be bacterial cell, it can be mammalian cell. What examples can we give? There is the Chinese hamster ovary. Okay, they are very commonly known as CHO cells. Okay. There is Chinese hamster ovary. There is NSO. There is BHK. There is HEC 293. Then there is C6. So these are mammalian cells. Okay. Then there can be yeast cells. And there can be mammalian cells. We can give another example, maybe the CAR T cell. T cells, okay. So they can be cells. Now these cells are used to produce biopharmaceuticals, which may be proteins. So bacterial cells, they have got a limitation in that they cannot do post-translational modification. Okay, so what is post-translational modification? After transcription, it gets translated. After that, there can be glycosylation. So glycosylation is a post-translational modification. So if it's a protein that needs to undergo post-translational modification, then bacterial cells would not be the choice. Then what would be your option? Your option would be mammalian cells. Okay, so they can do post-translational modification. So that is why we have different cells. So depending on the protein that we want to express, our choice of cells would be different. Tissues. Okay, so we can use maybe blood as an example. From blood, we have got blood products. Okay. We can also talk about maybe rabies anti-vaccine. Okay. Vaccine against rabies vaccine. Okay, that's produced in a horse. So again, the blood would contain the antibody, and it would be separated from the horse's blood. So yes, so blood is a tissue. So tissues can be used to produce biopharmaceuticals. What about organs? So if we have got, we can do a bit of engineering, and we can make transgenic goats, okay, which produce milk, and we can, using the techniques of biotechnology, we can express our therapeutic protein in the milk of the goat, and from which we can separate. Yeah. For example, human antithrombin. Can be expressed, and it can be isolated from transgenic goats. What about organism itself? So there is something called farming. Okay, farming with a PH. And farming is the production of biopharmaceuticals in plants or animals. is an example of hepatitis C vaccine being produced in potatoes. Again, there is lots of experimental work being done on animals. So if you need a drug, all you need to do is to consume the animal. If you want a vaccine, you can extract it from the plant. All right, so that is a bioprocess. So what are large molecules? They are biopharmaceuticals produced by a bioprocess. Molecular weight is greater than 10,000. Okay. So again, a biopharmaceutical is not produced by a pharmaceutical company. We call biopharmaceuticals are produced in a biotech company. Okay. So biotech companies usually produce biopharmaceuticals. And again, this is the bio originator. Okay. So analogous to the example of a factory that we saw, if there is a pharmaceutical industry that is producing the innovator biopharmaceutical, then that's a bio originator. Again, the patent life is 20 years. So after that, the patent expires. Okay, so if other companies jump in and try to produce the same kind of biopharmaceutical which is produced by a bio-originator, then those drugs which are produced by companies other than the innovator company, they are called biosimilars. Okay, so what are biosimilars? Basically, copies of the biopharmaceutical produced by the innovator company after the patent expires. So then the question is, why is it called a biosimilar? Can't we call it, why is it not called bio-identical? So the reason is because they are complex molecules and they are produced by a bioprocess which produces lots of variability. Okay. And so how does it produce the variability is what we are going to discuss right now. So let's take an example. Let us think of a biologic that is produced by a biotech company. What example shall we say? Let's say this is let's say this is a biotech company and this company is trying to produce insulin. Okay, so insulin is it's a protein. Okay. So what are the steps that this company uses to produce insulin is what we are going to see. So insulin, let's draw structure of insulin. So insulin is basically a dipeptide made up of an A chain. Let's call it A chain, and another chain called a B chain. Okay. This A chain is made up of 21 amino acids, and the B chain is made up of 30 amino acids. So, so the total number of amino acids for insulin is 51 amino acids. These two chains are linked by disulfide bonds. Okay, so this company is trying to produce insulin. Okay. So this is the goal of the company, to produce insulin. So normally in the body, insulin is produced as pre-proinsulin. Okay. So what is pre-proinsulin? So that's because it's a peptide, it's got a carboxy terminus and an amino terminus. Okay. And let's say this is a disulfide bond. Okay. That is the sequence like that. Okay. So this is called pre-pro insulin. Okay, it has got an extra sequence here, which is called the signal peptide. Okay, the signal peptide is 24 amino acids. Okay, so in a protein sequence, the number of amino acids, we call it residues. So there are 24 residues of the signal peptide. And then you have something called a C peptide. Now the C peptide links the A chain to the B chain. So A chain, we know it's 21 residues, and B chain is 30 residues, right? The C peptide has got 35 residues. So C peptide has 35 residues. So pre-proinsulin, so totally it would be 110 amino acids. Okay, so this is pre-proinsulin. So if we remove this part, the signal peptide, and we remove this part, then pre-proinsulin is converted to proinsulin. Okay, so what is proinsulin? Proinsulin is pre-proinsulin minus the signal peptide and obviously the amino terminus and the carboxy terminus. Okay, so that is proinsulin. So what they do is, this company, this biotech company, tries to find out this sequence of proinsulin. Okay. So it's a chain of 110 amino acids. I mean, um, the uh, yeah. So you take 110 amino acids. Okay, so 110 amino acids is the length of the proinsulin. Okay, so let's say this is 110 amino acids. Let's say this is the first amino acid here that we're going to call methionine. Okay. So at this end, let's call it the NH2 end, and we can call it the COOH end. Okay. First amino acid here is, let's call amino acid, the first one, it is methionine. Okay. Now let's try to draw the messenger RNA. Okay. So what happens is, on the messenger RNA, there is the, what is that called? If you said codon, you're absolutely right. This codon has got an anticodon. This anticodon is connected to an amino acid, and that amino acid is methionine. Okay, so this is the messenger RNA. Okay, so the anticodon for methionine is UAC. Okay, and the corresponding codon would be A, adenine uracil, AUG. Okay, but this is messenger RNA. Okay, and now if we draw the template DNA, would be the template DNA. It would be, because it is messenger RNA, there is U. Okay, otherwise it will be T, no? Right. So it would be T. If it is T, then it would be A. Okay, and guanine cytosine. Okay, so that would be the sequence on the template DNA. So then what would be the sequence on the coding strand? It would be T, complementary would be A, adenine, thymine, cytosine, guanine. Okay, so this would be the coding strand. Okay, so what has been done from the amino acid sequence? We have generated the coding strand, coding region. Okay, so now what the company is going to do is, would put this coding strand into what we call the vector, okay, which is usually a plasmid. Okay, so now if you think about this, there is no post-translational modification of insulin here. Okay, so the idea is this vector can be therefore introduced into a bacterial host. Okay, this process is called transformation, insertion of the vector into the host. So this is called transformation. Okay. So the next step is this transformed bacterial host is selected. Select transformed bacteria. Okay. So there are different techniques that we use to select the transformed bacteria. So the company also does the same thing. They select the transformed bacteria. And then they expand it, meaning they culture it. Okay, so initially they make pilot studies, and then they scale up. Okay, so what is pilot study? It is done in the lab. What is scale up? When they produce it in the biotech company. Okay, so first they do pilot studies and they scale it up. So when they scale it up, they try to grow it in something called a bioreactor. Okay, then the bacteria are grown in a bioreactor. And what does the bioreactor contain? Bioreactor contains media for the growth of the organism. Again, then it also supplies the appropriate oxygenation, temperature conditions, agitation, aeration. All those things happen in the bioreactor. Then what happens? It grows to confluence. So if we look at the growth curve of a bacteria in a new environment, there is initially a slight growth like that, and then it reaches exponential growth. So this is with time, and this is the biomass, number of bacteria. So initially it gets adjusted to the new media. Then there is exponential growth followed by a flattening of the growth curve. So when it reaches somewhere here, the bioreactor, it is switched off, or the growth is stopped. Okay. So after stoppage, what happens? The purification step begins. Okay, so there will be separation of the biomass from the liquid media. We use techniques like centrifugation. Okay, and then the bacteria is lysed, and then the desired product is separated, purified, assayed, then mixed with excipients and dispensed. And after quality control test, it is released into the market. Okay. So after purification, all the steps are done. Okay. So these are the steps of purification, and after that, it will be mixed. It will be, you get the idea. It is dispensed and sent into the market. So the point is, these steps, okay, so we are talking about a situation when the patent has expired. patent of the innovator has expired. Okay, so the company that is going to produce it, they can produce it 100 and they can file the application about 180 days before expiry of the patent. Okay, they can file for a biosimilar application. Okay, they can file it to be produced as a biosimilar drug. Now think about it. They are filing it 180 days before the expiry of the patent. Okay, so that means the innovator company still has got undisclosed data, undisclosed information. Okay, so which are proprietary, meaning only the company has got the information. But another company is now trying to produce it without this information. Okay. So there is undisclosed information about what are the constituents of the media. Okay. What are the exact conditions of growth, the optimized temperature, all the operating conditions. So we call it critical quality attributes. Okay. Critical quality attributes. Okay, so what are critical quality attributes? These are the processes that can affect the safety and efficacy of the product. Then there are key quality attributes. Okay. Key quality attributes. So what are key quality attributes? These are conditions which don't affect the safety and efficacy, but still it has got some influence on the formulation. So the company that is trying to produce the biosimilar product won't have all this information. Okay, may not have all this information, and it is produced in a bacterial strain. Okay, because we have taken the example of insulin. Okay, so which strain exactly? Okay, it's not known. Okay, the information about the pilot studies may not be complete. The issues related with scale up may not be fully disclosed. So the company that is going to produce a biosimilar drug has to do all these things, which brings about a variability in the final product. Okay. So that is the reason why a biosimilar is not bio-identical. Okay, so that's the reason why a biosimilar is not bio-identical. Okay, which brings us to two terms. So after all this, let's say the biosimilar gets approved. Okay. So the biosimilar, from the definition, it means it is similar. Okay. Not exactly same as the bio-innovator molecule. Okay. But it does not have any significant or clinically relevant difference from the bio-innovator molecule. But it is not bio-identical. Okay, which means a generic drug, like a generic drug, it cannot be substituted at the level of the pharmacy without the concurrence of the prescriber. Okay, it cannot be substituted without the concurrence of the prescriber. So that is why if the biosimilar drug undergoes further testing to show that there is no toxicity of substituting, and if more than one dose is given and then the biosimilar is switched with the bio-innovator molecule or vice versa, and that information is sent to the FDA, only then the biosimilar has got the potential to become an interchangeable drug. Okay, so are all biosimilars interchangeable? No. Only the ones which have undergone further testing are interchangeable. Okay. Okay, so that is the difference from generic drugs. Okay, so generic drugs can be substituted in the pharmacy, but biosimilar drugs cannot be. Only if they are bio-identical. Okay, so this is ideally what the FDA is trying to tell us. Okay, so how do we know where to find information? Where to find this information, whether the biosimilar is bio-identical and it can be interchanged. There is something called purple book. Purple book. So purple book is the book or the database of all the biological drugs which have been licensed, and it also contains information of biosimilars and interchangeable drugs. Okay, so that is the official source. What about generic? So in the generic drug, a similar book is called the Orange Book. Okay, so what does the Orange Book contain? It contains the bioequivalence data of all the generic preparations which are available. Okay, and so that can be referred for switching the drug at the level of the pharmacy. At the level of the pharmacy for the biologic drug, it is the purple book. Okay. So up till now we have talked about a lot about generic drugs. We have talked about biosimilar drugs. We have talked about why biosimilars are not bio-identical. Okay, so now let's look at official definitions. Okay, let's insert a slide. Okay, so this is the out here. Okay, and then let's try to zoom in. Okay, so this is the official definition of a biological product. So we said that biosimilars are copies of large molecule drugs. Copies of large molecule drugs. And these large molecule drugs are biologicals. Okay, so we have the definition. What is a biological product? So it may be a virus. Think about an active vaccine. Say the Sabin polio vaccine. Okay, so the killed one is Salk and the live one is Sabin. So an active virus can be therapeutic serum, say rabies antibodies. Okay, rabies antibody, toxin, diphtheria toxin. Okay, can be antitoxin again, anti-rabies. Okay, it can be vaccine, or it can be blood component, okay, or a derivative, allergenic products. Okay, so allergenic products, they can be used for desensitization therapies. Desensitization. Okay, so that is an immunological drug that is used to reduce a person's allergy. Okay, or arsphenamine or derivatives of arsphenamine. Okay, so this is the definition of a biological product. So here a protein is defined as anything that has got greater than 40 amino acids. Okay. So we said a biological product is produced by a bioprocess, and the bioprocess is producing it in a living system. But the official line is even chemically synthesized polypeptides greater than 40 amino acids is a biological product. So that is an exception. Okay, exception of the definition that we talked about. Okay, but that is applicable only to polypeptides. Okay. Let's look at the official definition of a biosimilar. Okay, it should be highly similar. Okay, they are not telling that it should be. They are not telling it should be identical. It should be similar to the reference product. What is the reference product? It is the product produced by the bio-innovator. Okay, or the innovator company that produced it first. So biosimilars are always compared to the reference product. Now it says it is accepted that there are minor differences in clinically inactive components. Okay, but the thing to remember is there is no clinically meaningful difference between the reference product and the biological products in terms of what? In terms of safety, in terms of purity and potency of the product. Okay, so there is no difference between a biosimilar product and a bio-innovator, or the trade name product in terms of safety, purity and potency. Okay, so a healthcare provider with a great healthcare prescriber with a great deal of confidence can use his judgment to use a biosimilar drug in a patient having a generic name product. Okay, but mind you, it is not bio-identical. Okay, So now we come to what are the examples of a biosimilar product. So let's look at some examples. Let's write some examples. Let's look at some examples. Okay. So adalimumab, okay, monoclonal antibody, and its name is, okay, we'll come back why it is named like this. Then trastuzumab, again it's a monoclonal antibody, and QYYP. Okay. Then there is filgrastim. Okay, so these are all representative examples. Infliximab. Okay, that has got a suffix DYGB. And bevacizumab, that has got a suffix BVZR. Rituximab got a suffix of ABBS. Okay, so these are the examples of biosimilars. Now, this is the equivalent of a generic name. Okay, and that equivalent generic name should have a suffix of 4 alphabets in this small letter. Okay, so this is the requirement for naming. Okay, so that is how biosimilars are named. Okay, so that is the requirement. Okay, so in generics we said the company had to prove bioequivalence. Okay, but in case of biosimilars, the company has to prove similarity to the reference product What is the reference drug? Reference drug is nothing but the bio-innovator molecule. Okay, similarity to the reference drug in terms of pharmacokinetics. Okay. Pharmacodynamics. Terms of their immunogenicity, efficacy, safety. Okay, with the bio innovator or the bio originator drug. Okay. So in case of generic, they need not do all these tests. Okay. So this is the abbreviated licensing pathway. It is not the full range of test that a bio-innovator molecule undergoes. It is less than that, but still it is more than the um, tests that need to be done for generic drugs. Okay. So that is again a difference between biosimilars and generics. Okay. So what have we covered? So what was our goal today? Our goal today was to talk about biosimilars, and we have basically discussed about generic drugs, and we said that what are generic drugs? They are small molecules. Okay, we defined what are small molecules? You're right, less than 1,000 daltons. And generic drugs, and we have talked about the differences between generic drugs and biosimilars. We've talked about biologics. What are the other names for biologics? Biotech drugs, technology derived drugs. They are called follow-on biologics. Okay, so they're basically biopharmaceuticals. Okay. Biopharmaceuticals. And then we talked about biosimilars. And we have said that they are not bio-identical. They cannot be interchanged. And we talked about lots of aspects of biosimilar drugs. There is a term currently making rounds called bio-betters. Okay. So from a regulatory aspect, there is no definition for a bio-better. So this is what the marketing, the companies that are marketing, they are using this term, bio-betters. Okay. From a regulatory standpoint, it doesn't have much of a meaning. Okay. So with that, let us conclude our discussion on biosimilars.